So this is chapter 6, bones and skeletal tissues. This is not going to be like lab where we just named bones. Basically what we're going to go through is how bone forms, how bone repairs, and a few types of bone breaks and bone disease. So the first thing to say is that ossification or osteogenesis is the process of bone formation. Formation of the bony skeleton begins in about month 2 of development. And one thing I want to talk about right away and be clear about is that generally hyaline cartilage precedes bone. So basically, you usually have some sort of hyaline cartilage template, and this is then ossified to form bone tissue. So this formation of the bony skeleton begins in about month 2 of development, and bone growth occurs until early adulthood when adult size is reached. But bone modeling and repair are actually lifelong processes, and we'll get into that in a little bit. Okay, so the first thing to say is that until about week 8, the fibrous membranes and the hyaline cartilage of fetal skeleton get replaced with bone tissue. This is something that's called endochondral ossification, and in this type of ossification, and again, to ossify means to form bone. In this, bone forms by replacing hyaline cartilage. And so when you look at the word endochondral, chondral or chondro refers to cartilage, so endochondral literally means within cartilage. So these types of bones are called cartilage or endochondral bones, and basically this ossification begins at a primary ossification center in the center of the shaft or diaphysis of the bone. And these types of bones actually form most of the skeleton. So let's take a look at this process, or we'll look at this process in a minute. The second type of ossification is called intramembranous ossification. And in this, bone actually develops from a fibrous membrane, not from hyaline cartilage. And so because they develop from a membrane instead of from cartilage, these bones are called membrane bones. So now let's take a look at endochondral ossification. In the first step, a bone collar forms around the diaphysis of the hyaline cartilage model. And if you look at this little picture, the hyaline cartilage is in blue, and you see in tan around it a bone collar forming, and in the bracket there's a primary ossification center where you're going to see the formation of bone tissue. In the second step, cartilage will calcify in the center of the diaphysis, so it will actually take on cartilage, and at that point it'll begin to deteriorate and form cavities. So now at this primary ossification center, you haven't just gotten a little bit of bone tissue and calcium in here, you've also gotten an open space. In the third step, you have what's called a periosteal bud, so an invasion of the periosteum, which is the fibrous outer membrane of a bone. This will invade this internal cavity, and you'll start to see the formation of spongy bone forming. And as well, you can see this is basically how the interior of a bone gets vascularized, because you see that there's the blood vessel of the periosteal bud that enters the bone. Some of the smaller foramina that we saw in lab are artifacts or leftovers of where this blood vessel invasion happened. And so these get called nutrient foramina, which are where this blood vessel actually invades and then stays. In the fourth step, you're going to see the diaphysis elongating. So the shaft of the bone elongates, and this internal cavity, which is now called the medullary cavity, begins to form. Also, you're going to see secondary ossification centers appear in the epiphyses or the ends of the bones. And so these will be invaded by their own blood vessels, and they'll also form spongy bone out there. In your final step, the epiphyses will ossify, and when this ossification is complete, you're only going to see cartilage remaining in the epiphyseal plates, which are known commonly as growth plates, and the articular cartilages. Now we'll take a closer look at these epiphyseal plates a little later in the PowerPoint. So the second type of ossification, which is intramembranous ossification, this begins with again, not cartilage, but fibrous connective tissue. These membranes get formed by mesenchymal cells. And these will form both the clavicle and the major cranial bones, the frontal, parietal, occipital, and temporal bones. And in this type of ossification, there are four major steps. First of all, you see the formation of ossification centers when these mesenchymal cells cluster, and they'll differentiate into osteoblasts. So the name of this cell, osteoblast, remember that a blast is a type of cell that transforms into or forms something, and osteo refers to bone. So an osteoblast is going to be a cell that actually forms bone matrix. At this point, osteoid, which is non-calcified bone matrix, it gets secreted and then it gets calcified. In the third step, a type of bone called woven bone gets formed when you lay osteoid down around blood vessels, and this will result in the typical structure of spongy bone called trabeculae. So it's the outer layer of this woven bone which forms the periosteum in these membranous bones. In the fourth step, lamellar bone replaces woven bone. So lamellar bone is actually going to be bone with lamellae. And if you'll recall, these are these rings of bone matrix that surround either osteons, or in this case, are in the trabeculae of spongy bone. So once these lamellae replace woven bone, you'll see red marrow appear afterwards. So here's our four steps. This is ossification centers developing in the fibrous connective tissue membrane. So you see the mesenchymal cells which have clustered, collagen fibers surrounding, and in this ossification center, this is where you're seeing some of the differentiation into osteoblasts. Again, the osteoid is non-calcified bone matrix, and the osteoblasts themselves are the cells that will continue to produce the bone matrix. In the second step, osteoid gets secreted and then calcifies. So again, you see the osteoblasts there secreting osteoid. The osteoblasts, which are trapped within this osteoid, within this calcified osteoid, become osteocytes. And so osteocytes, if you'll recall, are the cells that are within lacunae in bone tissue. And these are the cells that don't produce bone matrix, but actually maintain the bone matrix. And again, you see the newly calcified bone matrix surrounding the osteocyte. In the third step, you see the formation of immature spongy bone and periosteum. And so as the osteoid gets laid down between these blood vessels, you're going to get a honeycomb of immature or woven spongy bone. And so you'll see the mesenchyme up at the top level. It'll actually condense to form the periosteum. And so you can see that it's vascularized. It'll condense on the external face of the bone. It becomes the periosteum. The second label down is the trabeculae of the immature spongy bone, and then of course you see the blood vessel. In the fourth step, you can see how this develops. So you're going to see compact bone replacing immature spongy bone just deep to the periosteum, but deep to that compact bone, you're still going to see trabeculae. Okay, so the trabeculae that are just deep to the periosteum get remodeled and replaced with compact bone. So again, this is the type of bone that's got osteons and so forth. In the center, you got the immature spongy bone being replaced with mature spongy bone. And so this is where you see the formation of trabeculae and all. And the space eventually gets filled in with red marrow. So here we see the fibrous periosteum, the osteoblast, the compact bone, and the diploe, which is spongy bone cavities that contain red marrow. Okay. That's the initial formation of bones. Now, once they're formed, how do the long bones actually grow? Well, this is something that's called interstitial growth, and this requires epiphyseal cartilage in the epiphyseal plate. Now, this plate maintains a constant thickness, and so the way that this plate maintains constant thickness is that on the outer edge, or the growing edge, I should say the leading edge of the epiphyseal plate, you've got cartilage growth. And as this cartilage grows, it'll actually push the tissue on the leading edge outwards. And on the lagging edge, which would be the edge that is following the epiphyseal plate, you're going to see bone replacement on this edge. And so the epiphyseal plate consists of 5 zones, which we'll see in the next slide, or 2 slides from now. One zone is the resting zone, and this is an area of cartilage that's relatively inactive. So there's not any kind of growth or matrix production going on in this zone. In the second zone, you've got what's called the proliferation or growth zone. And so this is, again, sort of on the leading edge of the epiphyseal plate. And this is an area of cartilage that has cells that are rapidly dividing. Now, as these cells move upward, they're actually going to push the epiphyses away from the diaphysis, and this is what causes lengthening of the bone. The third zone is called the hypertrophic zone, and so in this case, the cartilage lacunae are going to enlarge and erode, and so hypertrophic basically means larger form, and basically these older chondrocytes are closer to the diaphysis, so they're not on that leading growth edge. And again, here the lacunae are going to enlarge, and they're going to erode, and they're going to start interconnecting. The fourth zone is the calcification zone, and in this zone, the cartilage matrix is beginning to calcify, and at the same time, the chondrocytes will die and they'll deteriorate. And the last zone is the ossification zone. So what the chondrocyte deterioration leaves behind is these long sort of little spikes of calcified cartilage. So these get eroded by osteoclasts, and so osteoclasts are the type of bone cell that breaks down bone tissue. So they're always going to be involved in eroding or deteriorating bone tissue. So once osteoclasts erode these spicules, then osteoblasts are going to cover these spaces in new bone. Ultimately, this gets replaced by a spongy bone, and again, as these spicules erode, you're going to see an enlargement of the medullary cavity. So let's take a look at a diagram. So now we're going to take a look at an actual picture of an epiphyseal plate. So on the left, you're taking a look at a photomicrograph. So this is not an electron microscopy picture. This is a light microscopy picture. And on the right, it's a diagram, a sort of a cartoon depiction of these actual zones. So let's take a look at these different zones. Up at the top, you see the resting zone. There's not really a lot going on here. Not a lot of cell division, not a lot of action going on. In this zone, the proliferation zone, this is where you see the chondrocytes or cartilage cells undergoing rapid mitosis. And it's this growth that's actually going to push the epiphyses away from the diaphysis. This really is the force behind lengthening of bones. Below that, you'll see the hypertrophic zone. And if you'll notice, again, this is the zone in that bracket. In this zone, you see the enlargement of the lacunae here and the enlargement of the cartilage cells themselves. In the next zone down, you see the calcification zone. And what I want you to notice about this is that you're actually seeing some of those spicules that are left over by the dying cartilage cells. And so that's the reason why those lacunae look like they got little lines in them. And so in these spaces, the matrix is becoming calcified and the matrix is deteriorating, and this is the reason why generally the lacunae are larger than they are in the hypertrophic zone. The last zone is the ossification zone, and in this zone, new bone is forming. So these are the different zones of the epiphyseal plate. Now, if you'll take a look on the very left, you have an X-ray image of the right knee. And so on the bottom, this is the zone that's actually being depicted. You've got the proximal epiphyseal plate of the tibia. This is enlarged. Now, the epiphyseal plate above it is on the femur, and what you would see in the femoral epiphyseal plate, because it's a distal plate, you'd have to flip these images upside down. So you would see the resting zone on the bottom and the ossification zone on the top, because the growth of the femur, in this case, in the distal epiphysis, is not in a proximal direction, it's in a distal direction. Now, an experienced pediatrician or radiologist can often tell from the width of these epiphyseal plates how much more growing that an individual has to do. So what happens when an individual stops growing is that the proliferation zone slows down in terms of the rate of mitosis. So that mitosis slows down. And what that allows is for the ossification zone to catch up with the proliferation zone. And what you'll see is the proliferation zone shrink and shrink and shrink and shrink until it becomes completely ossified. And at that point, you're actually going to call this the epiphyseal line, not the epiphyseal plate. So when you see the complete ossification of the epiphyseal plate, someone is completely done growing. So near the end of adolescence, as I've just covered in the previous slide, sorry, chondroblasts divide more often. The epiphyseal plate thins. It closes when the epiphysis and the diaphysis fuse. And usually bone lengthening ceases in females around 18 years of age and in males around 21 years of age. Now, this is not exact. This is an average number. So you will see sometimes growth occurring a little bit longer, sometimes growth ceasing before then. I think we all know that person who got out to a big lead and was the tallest person in 6th grade and then was the shortest person as a senior in high school. So the numbers on when this happens vary from person to person. Okay, there is some hormonal regulation of bone growth, and the hormones are as follows. So growth hormone is your most important hormone when it comes to stimulating the activities of the epiphyseal plates in infancy and in childhood. Thyroid hormone actually sort of controls or modulates the activity of the growth hormones, and what this is supposed to do is to ensure proper proportions. Now, at puberty, testosterone in males and estrogens in females will actually act synergistically with growth hormone. So what they'll do is to accelerate the rate of growth, and this is what actually causes the growth spurts that you see in adolescence. But these two hormones will also end growth by inducing the closure of these epiphyseal plates. So it can be really unfortunate. Excesses or deficits of any of these hormones can cause abnormal skeletal growth. So you can see what's called gigantism with an excess of growth hormone. So this is where people will grow extremely tall. And you can also see something called dwarfism, in which you have sometimes this is caused genetically, and sometimes this is caused with a hormonal deficit. And in what's called pituitary dwarfism, you see someone with more or less normal proportions, but they're just smaller. Okay, now once you're done growing, this is not it for the activity of bone tissue. So bone remodeling occurs on a constant basis. So it's about 5 to 7% of bone mass that gets recycled each week. And what that means is the spongy bone in your skeleton gets completely replaced about every 3 or 4 years, and the compact bone gets replaced about every 10 years. And so one of the strange things that I think might happen with, say, my skeleton in particular, I've lost a lot of weight over the past 5 years. So my guess would be that if I died and was skeletonized, that people would probably say that I was still pretty big, because my skeleton hasn't had a chance to catch up with reduced weight. So I would look like I had a fat person skeleton in a, moderately shaped person. So bone remodeling is going to consist of both bone deposit and bone resorption. And both of these processes occur at the periosteum, which is that outer membrane of the bone, and the endosteum, which is the inner membrane of the bone that lines the medullary cavity. So it consists of remodeling units, and so you've got adjacent osteoblasts, which are responsible for bone deposit, and osteoclasts, which are responsible for bone breakdown or resorption. These groups of cells will actually coordinate the remodeling process. And so resorption is the function of osteoclasts. They're going to dig depressions or grooves as they break down matrix. And the way that they break down matrix is to secrete lysosomal enzymes and actually protons. And protons are acid, and this is going to digest matrix. This acidity will convert the calcium salts that are present in bone tissue to soluble calcium. So basically, the calcium is dissolved into the bloodstream. So even though remodeling occurs continuously, it is regulated by genetic factors and a couple of different control loops. And so those control loops are, first of all, hormonal controls. And basically, you've got negative feedback loops that control blood calcium levels. Now, blood calcium, as you've actually seen in skeletal muscle physiology and as you'll continue to see in nervous system physiology, calcium functions in a lot of processes: nerve transmission, muscle contraction, blood clotting, gland and nerve secretion, cell division. So calcium is an extremely important mineral to be present in interstitial fluid. And so if those levels go too high or go too low, you're going to have some sort of control mechanism for them. Now, 99% of the calcium that you have in the body is found in the bone. If you need to absorb new calcium, there's definitely a requirement for vitamin D. So you may recall that anytime that you see a gallon of milk, you rarely see something just labeled as milk. You'll see vitamin D milk. And the reason why is because the vitamin D is necessary to absorb the calcium that's actually in the milk. It makes the milk more nutritious. The second control loop is response to mechanical stress. So we'll take a look at that later on. So other hormonal controls, this is controlled by a number of different hormones. So the first is parathyroid hormone. This is produced when you've got low blood calcium levels. And what this does is to cause osteoclasts to resorb bone. So if you've got low blood calcium levels, then your osteoclasts will dissolve bone and release the calcium into the bloodstream, which raises your levels back up to normal. Now, once you reach the normal level, PTH secretion will actually stop. Calcitonin is produced in response to high blood calcium. Now, if you give this to someone at high pharmacological doses, you can lower blood calcium levels temporarily, but under normal circumstances, the effects are a little bit negligible. Now, if you want to remember which of these does which, then you can think of calcitonin tones down calcium. So this is what will be produced in response to high blood calcium levels. Now, calcium homeostasis is a narrow range because even really small changes in blood calcium levels can cause a lot of neuromuscular problems. Hypocalcemia, which is low levels of calcium, this can cause hyperexcitability of the membranes that are involved. And as you've seen from the muscle chapter, excitability of the membranes, this basically would mean that action potentials can start a whole lot more quickly. And this is something that is a real dysfunction in both nervous tissue and in skeletal. Can lead to a formation of calcium kidney stones. So the second type of remodeling is response to mechanical stresses, and this is the idea that bones reflect stresses that they encounter. So basically, bones get stressed when weight bears on them or when muscles pull on them. And there's a law called Wolff's Law that says that bones will grow or remodel in response to the demands that are placed on them. Now, stress, as we'll see in the next slide, is usually off-center, and bones will tend to bend. And what this bending force does is to compress one side and stretch the other side. And you're going to see the diaphysis, or the shaft of the bone, the thickest where the bending stresses are actually the greatest. Now, even though there's this compression and stretching that's placed on the bone can actually be hollow because these two forces will cancel each other out in the center of the bone. And when you see the diagram on the next page, I think that'll make a little bit more sense. So here, if you'll look at the stress that's placed on the bone, if you look on the left, there's tension at that point, because if you look at the purple arrow up at the top, you've got a lot of load that's on the femur at this point. So basically, at the dotted line, this is where the bone is threatening to be bent. So on the left, you're going to see tension towards the outside of the bone, which is going to kind of try to stretch out the bone. And then on the right side, you're going to see compression where the bone is kind of getting squashed. Now, those two forces cancel out at the point of no stress. That's indicated by that dot. And because those forces cancel one another out, this is the whole reason why you can afford to have a medullary cavity at all. Because at that point, you don't need as much bone material to keep the bone together. Now, Wolff's law will also explain some things that happen that you can see in the skeleton. Whether you're right-handed or left-handed, this will result in thicker and stronger bone of your dominant limb. And this is something that we talked about in lab. Curved bones are the thickest where they're most likely to buckle. You're going to see trabeculae reinforce or form trusses along lines of stress. So in spongy bone, the trabeculae are not laid down at random. They're actually going to be directional along lines of stress. And finally, you're going to see large projections occurring where you have heavy and active muscles attaching. So someone, say a weightlifter, they're going to have a lot of thickenings at muscle attachment sites where the muscles are that they use the most. If someone has a really difficult physical labor job, you're going to see the same thing happen. And so a forensic anthropologist can actually tell whether someone was heavily muscular or not, depending on how large that these projections actually get. Now, the opposite side of the spectrum is the bones of fetuses and bedridden people. These, I wouldn't call them quite featureless, but the projections are really reduced because the bones have not been put under stress. Okay, so the next thing that we're going to talk about are injuries and bone diseases. And so we'll start out with fractures, and there are three sort of either or fracture classifications. And so where are the position of the bone ends after the fracture? Are they together or have they been separated? And so in non-displaced fractures, although the bone has been completely broken through, you don't see any displacement of the ends of the bones. In displaced fractures, you're going to see these ends come out of their normal alignment. The second thing is the completeness of the break. So a complete fracture is when the bone is broken all the way through, and an incomplete fracture is when it's not broken all the way through. So is the skin penetrated? Now, I want to point this out. You have to have both a displaced fracture and a complete fracture for skin to be penetrated. And so in an open or compound fracture, the skin actually gets penetrated. In a closed or simple fracture, although there may be a complete break and there may be displacement, the skin is not penetrated. Fractures can also be described by their location, by their external appearance, and by the nature of the break. And so we'll go over just a few different examples of this. There are tons of different names for different fractures. So fractures of a lot of different bones get their own names. And if you want to see a really long list, go look up fractures on Wikipedia. It's got a pretty complete and pretty astounding list. So what are some of the common types of fractures? A spiral fracture is when you have twisting forces applied to a bone. This is pretty common in sports. And if you can think of a scenario, say where somebody plants and plants a cleat in the ground and the cleats get stuck as their legs twist, this is something that can happen to an athlete at that point. This is an epiphyseal fracture. Now this is only going to occur to someone who is not through growing yet, and this is when the epiphysis separates from the diaphysis at that epiphyseal plate. This tends to occur where the cartilage cells are dying, so this is going to happen at that hypertrophic zone that we saw and where the calcification of the matrix is occurring. So at that point, that's a weak point of the epiphyseal plate, and you can get an epiphyseal fracture there. So repair is going to involve four major stages. The first is hematoma formation. The second is a fibrocartilaginous callus formation. So once again, here we see something going along with a common theme that cartilage precedes bone. The third thing is a bony callus formation, and so although this will be bone tissue, this is not something that's going to be as strong as mature bone tissue. You're going to have bone remodeling occur for that to happen. So let's take a look. In the first stage, you see a hematoma. So this is basically a big blood clot. In the second stage, you see a fibrocartilaginous callus forming. So at this point, you're still going to be in a cast. The bone is still going to be very weak. You're going to still going to have a lot of pain when you try to put any sort of weight on this, but at least your hematoma has been replaced by some sort of connective tissue. In the third stage, you see a bony callus forming, and this is where healing is fairly close to complete, but not quite. So although you've got bone tissue all the way through, at the point of the break, this bony callus is actually going to be weaker than the bone tissue surrounding it. This is where you can have a re-break that possible. And in the fourth case, you see bone remodeling occurring. And ironically enough, when you break a bone, when the fracture heals back, it'll actually heal back with a higher proportion of compact bone, so that the point of the break is actually stronger than what it was before the injury. So if you've ever had a sprain, an ankle sprain, you might have heard the doctor say you'd have been better off breaking it. And that's absolutely right, because when you tear tendons or ligaments, it's very tough for them to grow back because they've got a really poor blood supply. When you break bone, it will heal, and it heals faster than tendons or ligaments because it's got a much better blood supply, but it also heals back more strongly. So the last thing that we're going to look at in this PowerPoint is actually bone disorders. And so you can have imbalances between bone deposit and bone resorption. And these are basically what underlie just about every skeletal disease. And the three major bone diseases that we're going to cover are osteomalacia and rickets, osteoporosis, and Patch disease. So first up, osteomalacia occurs when bones are poorly mineralized. And in this case, osteoid, so the bone matrix, gets produced, but without enough calcium, you don't get the deposition of calcium salts. And this results in bones that are softer and weaker, and you can have some pretty considerable pain upon bearing weight. If you see this in children, it's going to be called rickets. And unfortunately, this can result in permanently bowed legs and other bone deformities. At this point, the bone ends are enlarged, the bones are abnormally long, and the diaphyses are a little bit thinner. And again, this is going to result in bowed legs. The cause of all of these is either insufficient calcium, or it could be vitamin D deficiency. So remember that even if you're getting enough or maybe even too much dietary calcium, without an adequate amount of vitamin D, you're not going to get the absorption of vitamin D from the GI tract. So this is a picture of osteoporosis, which happens most often in aged and postmenopausal women. So up at the top, you see trabeculae of normal bone, and at the bottom, you see osteoporotic bone. And as you can see, there are holes in the bones. Osteoporosis literally meaning porous bones. So the reason why this affects 30% of women aged 60 to 70 and 70% of women by age 80 is because estrogen actually plays a role in bone density. What estrogen will do is actually cut down on the activity of osteoclasts. So when estrogen is no longer being produced at higher levels, osteoclasts will have more activity, and you'll actually see these pores form in the bone. Now, men are less prone to osteoporosis because testosterone in men has the same sort of protection that estrogen does in women, but men don't have that radical reduction in their sex hormone when they become older. So there are additional risk factors for osteoporosis. Insufficient exercise that stresses bones. So even though your bones get placed under stress, this is a good thing for them, because if they're not placed under adequate stress, you're going to see osteoporosis. So this is something that astronauts are actually at a risk factor for, unless they work out as heavily as they can with weights. And even then, they do see reductions in their bone tissue, and they do see at least mild osteoporosis. Diets that are poor in calcium and protein can also result in this. Smoking is a risk factor for osteoporosis. Some people are more prone to others because of their genetics. Um, Hormone-related conditions can cause this. Hyperthyroidism can actually cause osteoporosis, as can diabetes mellitus. And this is what you would know as type 1 or type 2 diabetes. Um, Excessive consumption of alcohol and some medications can also cause osteoporosis. In Paget's disease, I want you to take a look at the X-ray on the top and the X-ray on the bottom. And what you'll see is pretty consistent bone density at the top, and at the bottom, you're actually going to see some portions of higher bone density, but a lot of portions of lower bone density. And so you're going to see excessive bone deposit on the areas that are brighter in the lower X-ray photograph. So differential bone deposit and resorption can cause bone to grow fast. It can develop poorly. This is something that's called Pagetic bone, and in this case, and as you can see in the picture, the bottom picture, even though you've got some bright spots, most of it's actually dimmer. And this is because you've got a high ratio of spongy to compact bone and less mineralization, less calcium. And I want you to remember that it's the minerals that actually show up on an X-ray. Now, this usually occurs in the spine, the pelvis, the femur, and in the skull. It rarely occurs before age 40, and the cause is unknown, but there is some evidence to suggest that it's possibly viral. Your treatment is going to include bisphosphonates and also calcitonin.